Good afternoon, ladies and gentlemen. My name is Peter Littleton. Welcome to the IFST webinar on utilising the past to predict the future, organised by the Food Safety Special Interest Group. I'm the honour of being the chair of the group at the moment, and also I'm UK Technical Director for Christain's Food Hygiene. We've organised this webinar to explore the importance of learning from the past and trying to learn from and not make the same mistakes twice. We're going to look at past incidents and focus predominantly on issues discovered through the analysis of food samples and how often they've resulted in new technical understanding. This webinar will be recorded and will be uploaded onto the IFST Webinars Hub after the session in about a week's time. Please feel free to make any comments, and if you wiggle your mouse and look on the chat bar at the bottom, the taskbar, you will see there is a Q&A function. So if you could submit all of your questions through that, and also you can upvote other questions, and we'll be utilizing those upvote rankings later on when we have the questions presented to our speakers. We will respond to as many of those questions as possible. As I'm sure you'd appreciate, sometimes we have more questions than there are time, and we'll explain at the end what we'll do with the questions that we're unable to get to at the end. So we have two fantastic speakers today. We have, lost my notes, sorry, Professor Louise Manning. She's Professor of Sustainable Agri-Food Systems at the University of Lincoln. And Louise has worked as an academic management consultant, lecturer and business and personal coach for over 35 years, addressing business management, QA, brand equity, food safety, animal welfare, corporate social responsibility, ethical issues and environmental protection. And our other speaker today is Michael Walker, Honorary Professor at the Institute for Global Food Security at Queen's University Belfast, and we'll be hearing from Michael first. Michael's an Honorary Fellow at the IFST and Honorary Professor, as I say, the Institute for Global Food Security in Queen's. He runs his own chemical legal consultancy, mainly providing expert witness services on chemical exposure. He retired as head of the Office of the Government Chemist at LGC at the end of March 2021, and he serves on our IFST Scientific Committee and the External Affairs Committee. We're also joined today by Charlotte Savage, the Head of Technical for the Bakery and Food Services at Sainsbury's. Charlotte's been at Sainsbury's for 6 years in various roles, and more recently has taken on the Head of Technical for bakery and food services. Before that, Charlotte worked in the supplier base for over 5 years, where she fell in love with everything technical and has never looked back. So Charlotte will be moderating our questions later, and we'll be presenting the questions that you put again through the Q&A button through to Michael and Louise. So with no further ado, and we'll get into the guts of the afternoon, I have pleasure in handing over to Michael Walker. And Michael, if you'd like to unmute, share your slides, and away we go.
Thank you very much, Peter, and thanks to IFST for inviting me to do this talk. It's based on some of my experiences when I was the referee analyst to the government chemist, as you can see. And it's really thoughts to prompt discussion around learning from the past. Now, I know we've got quite a few attendees. I don't know the individual circumstances of your work, whether it's research or an industry. So what I'd like you to do is think as we work our way through these case studies, how does that apply to me? How do those lessons learned apply to my situation? And I'd be pleased to hear your comments on that. So let me make sure I can move on. And yes, we have a quick look at the big challenges. You all know them, but we'll really drill down into technical detail quite quickly through sampling and analysis, the things that happen when nature springs a surprise, a little bit about bioinformatics, and then ending up on allergen analysis and reporting, which is a favorite topic of mine. Just to say a word of caution, This is about sampling and analysis in really chemical and bioanalytical laboratories. But there are many other ways to safeguard your product and your supply chain. And risk mitigation will be dealt with by Professor Louise Manning. And then we have some conclusions, and Charlotte will manage the questions. And some of the big risks, we will not be tackling these directly, but they may influence our discussion. And you see here geopolitical instability, climate change, nutrition-related morbidity and mortality, population growth, and you could add many others. And they give rise to the technical problems that you have to deal with, ranging from HR issues, can you get enough workforce to produce the goods, the food that you're producing, through to toxins, contaminants, and allergens, and all the nutritional cardiovascular disease, etc., which is occasioned by obesity. And on the other side of that coin, hunger and poverty in the world, which is another huge problem. We're going to start with a poll, and I'm told that I don't need to stop sharing my slides for this poll to come up. Oh, wow, there it is. It works. The audience poll, we're going to repeat this at the end of the session. So we've got about, what, 30 seconds or so. Which of these best assures the safe production, presentation, and consumption of food? And if you opt for one, please. It could be auditing the process, could be documenting the process, it could be testing the process parameters and final products, could be ensuring a robust prerequisite plan and full HACCP, and it could be training, awareness, and development of all food handlers. Now, there's no single right answer, but what's your preference among these options? Okay, interesting set of results. Testing comes way down the list. And the winner at the moment is a robust prerequisite plan and full HACCP. And I can't say I disagree with that. So there we have it. Auditing and documenting don't get any votes, but they're important. Testing gets very few votes. Nevertheless, we're going to talk about testing for the next 30 minutes or so. Now, the first thing that you think about when you're talking about analysis is sampling, how to do the sampling. And a good sampling plan is crucial, because analysis means nothing if the sample has been poorly taken, that isn't representative of the commodity you're trying to look at. And the example here is for aflatoxins. And aflatoxins, as you know, are genotoxic carcinogen, known for a long time. We've been analyzing for aflatoxins for a long time. They've been a problem for a long time. And they still are. So no apologies for using this as an example. And the other good reason that it's an example is that it's an exemplar of a contaminant that is inhomogeneously distributed in any lot, batch, or consignment. Because mold growth occurs at unpredictable and intermittent inhomogeneous places in a consignment, and therefore the secondary metabolite, that's the aflatoxins, particularly aflatoxin B1, is found at unknown, randomly distributed places in the consignment, and therefore the sampling has to mirror that. And there's been decades of work and hundreds of studies published on the operating characteristics of various sampling plans. And that's resulted in a statutory sampling plan in European and UK law. And it depends on taking a number of increments, and the number of increments, up to 100, the number is based on the size of the consignment. And those are bulked together. And depending again on the size of the consignment, they may be split into 2 or more manageable sublots. And those sublots themselves then are divided into three parts. One part goes to the official control laboratory, the public analyst laboratory. The second part goes to the owner of the consignment. The third part is kept by the official sampler, probably a report. And that third part is kept securely and can be sent to the government chemist if a dispute arises between the other two laboratories. And that's where I came in, and my successor now, Paul Hancock, is doing the same thing, and I believe that aflatoxin cases still arise. Then in the laboratory, this sample, it can be up to 20 to 30 kilograms in size, is thoroughly mixed, and we see here that being done in a new clean cement mixer. And then that mixed sample, and you can see it's peanuts in shell, is slurried with added water in a high shear mixer in order to homogenize the sample. And at that point, we know that that sample is as good as we can get with regard to homogeneity, and it can be subsampled then into the three parts for the prosecution, the defense, and the referee. And then it goes on for analysis. The analysis is pretty straightforward. It's been known for years. It's for aflatoxin B1 and the other aflatoxins, and for many of the mycotoxins, it's usually quite a straightforward liquid chromatography with, for example, the B1, G1, B2, G2, fluorescence detection. And it's not a particularly expensive setup. The things to remember are that in legislation, for good scientific reasons, a number of things have to be taken care of. We have to estimate our recovery, and we have to correct for recovery. And there are limits in which the recovery has to fall. We must also correct for the added water, because the added water we put in to slurry the sample isn't part of the sample itself. Therefore, we mathematically correct back to the sample itself. And if we're dealing with peanuts in shell, the limit applies to the edible portion. So we have to correct for the shell to core peanut ratio. So those are the things we have to look at. We also need to have a look at the expanded measurement uncertainty of the determination. And as you can see in the diagram on the left of your screens, if there's a maximum limit, only the measurement on the extreme left is definitively above the limit to the criminal burden of proof. And that is because the lower bound of the expanded measurement uncertainty is above the limit. And that's not the case for any of the other three measurements on the chart, despite the fact that the second one, the mean of the results, is above the limit. And then the third one, the upper bound is above the limit. It's only the first one which is used for non-compliance estimation. Here's an example. Could have picked any one from hundreds. This is pretty much at random. It was melon seeds. It was a two-part. So each of the three parts was itself a two-part sample. Such was the size of the assignment. The public analyst reports give results for each of the parts, and you can see there it's just over 3 micrograms per kilogram, and the associated measurement uncertainty is reported and is displayed. The laboratory operating for the food business operator reported results below the limit, but didn't indicate measurement uncertainty, nor did they indicate anything to do with recovery, slurry correction, etc. The government chemist, the team I was heading at the time, produced 2 other sets of results, and these are multiple analyses over multiple days with certified reference materials, and also confirmation of the molecular identity using LCA SMS. And you can see we confirmed the public analyst's findings, as we do, as the little pie chart shows, most of the time in relation to mycotoxin results. So why do we get disputes? And we get them in particular because a sample is taken in the country of export and is found to be compliant, but when the consignment sampled at the UK border, it's found to be non-compliant. And is that because of sampling at the country of export? Is it because of mold proliferation in transit? Could be either, but you need to investigate both if you're seeing a spate of that sort of scenario. It could also be, and this applies either to the laboratory in the country of origin or to the laboratory in the UK, because we've seen reports from UK laboratories display these probable errors, that the lab forgets about the nut to shell ratio, or forgets about the slurry ratio, or forgets about the recovery correction, or doesn't take into account measurement uncertainty, or any combination of or all of these. And so those are the things that we think you should look out for, and questions you might ask your lab. Now, there are occasions when a laboratory result is questioned, not because of anything the laboratory has forgotten about or done incorrectly, but because nature has sprung a nasty surprise. And the three examples I want to talk about are SEM, semicarbozone, I'll explain what that means in a moment, the almond mahalab allergen story, and then Manuka honey and stable carbon isotope ratio mass spectrometry for authenticity. And just as a random screenshot from the internet, here's completely unrelated, no aspersion cast against this particular brand. I just wanted to put it up so you can see that the price is £90. So these are not particularly trivial things to buy. But looking first at semicarbozone, this is to do with nitrofuran antibiotics, and they're prohibited for use in most food-producing animals, in all food-producing animals in most jurisdictions. And that's because of concerns about carcinogenicity and mutagenicity of the nitrofurans themselves and their metabolites in the animal and in humans. So they're still used and authorized in human and veterinary medicine for non-food producing animals. They're widely available. And they're rapidly metabolized. So in the animal, you'll not find the parent drug, and therefore there's no point in looking for the parent drug to distinguish, to find out if it's been illegally administered. What we need to do is look for the marker metabolites. And that works well apart from, for nitrofurazone, the semicarbozone. And that's the only viable marker metabolite that's been found so far. So first of all, it was found that, for reasons best known to the creator, if you believe in him, her, that that semicarbozide naturally occurs in the shells of shellfish, and that can happen without any tissue-bound SEM in the meat of non-treated animals. Then, in 2013, Glenn Kennedy from Northern Ireland, in the Veterinary Science Division, published a study that showed that some of the epidermal layer that synthesizes new shell remained with the meat, and SEM could migrate from that layer, that membrane, into the meat itself on the surface. And so what we have to do is excise the core of these animals. And this is a photograph, a lovely photograph of tiger prawns taken by my former colleague, Kirsten Gray. And it's a very labor-intensive, time-consuming process to excise the core flesh and then run the 2-day process to check for the presence of semicarbozide, tissue-bound semicarbozide in the core flesh. But that's the only way that you can prove definitively that the animal was treated illegally with one of these, with nitrofurazone. So why the shell manufacturer, why these things manufacture semicarbozide? I can only guess. It's to frustrate analytical chemists, and the creator has a sense of humor after all. And Karen Goodburn, who can't be with us unfortunately today, has put together a couple of these media stories from this going back to 2007, but the whole story started long before that. Australia, multiple antibiotics from Asia, and it's not over yet because Karen also looked at recent RACIFs, and everybody will know that Louise Manning is a doyen of the RACIFs. But look at this, we're seeing, still seeing problems and recalls of nitrofuran metabolites, mostly the not the one we'll be talking about, but bear in mind they still all could be there right up to a couple of months ago. The next story that I'd like to talk to you about is the almond and mahalab case, and this is something that happened a few years ago in relation to spices. Cumin and paprika were the spices we dealt with. Most people know the story now. A sample of cumin was taken routinely. It was found to contain almond, and annexed to legislated allergen, which must be declared if it's added to a food, or if it's present in dangerous quantities, which might be a risk for those with almond allergy. Now, the recall affected Bart's Ingredients, and Bart's Ingredients head of technical was Greg Corbishley at the time, and Greg won't mind me mentioning his name because he's often, this is something he talks about himself, and permission to recount what happened. Because Greg did a very clever thing. When the almond was found in the cumin, he sent off to the milling house in Turkey for a list of everything they handled. And he went through it line by line. And when he came to a thing called mahalab, he didn't know what it was. So he looked it up on the internet. And he had the good sense to realize that because it was Prunus mahalab and almond is Prunus dulcis, he thought, oh, same family. I wonder, is the ELISA cross-reacting? So he got some tests commissioned. They showed him that indeed 2 or 3 ELISAs that he got tested did cross-react to mahalab. Mahalab isn't on the Annex 2 list, therefore it's not a legislated allergen. Now that's the other thing that's interesting from this, is that the milling house in Turkey had no idea that anywhere on earth anybody would a problem arise for mahalab, because they didn't recognize it as an allergen either. Although sequence homology might give rise to problems. We can talk about that in the discussion. And so the FSA, who had initiated the recall, referred the matter to the government chemist. And we looked at the ELISAs, we looked at 3 different ELISAs. They all cross-reacted to different degrees. Then what we had to do was sort it out. Was it mahalab or was it almond? And what we did was to assemble a cross-disciplinary team of scientists. We had molecular biologists who looked at developing from a standing start a real-time PCR assay for mahalab, and then later on a PCR melt curve screening assay for the common Prunus species. We had protein mass spectrometrists look at LC-MS/MS for Prunus species to look for species-specific peptides, which would be characteristic for the proteins in either almond or mahalab, so that they can be distinguished. And these tests were all run. This work was done. Now, in the end, none of these approaches would have stood up in court alone, certainly not the ELISA, but neither the PCR or the LC-MS, and it's for various reasons, which I can explain if you're really interested later. But put together, the weight of evidence was overwhelming. But the cumin sample did not contain almond, but did contain Prunus mahalab. But a later and companion sample of paprika did contain quite a bit of almond. So that put everything to bed. We had 5 publications from that investigation. It took 8 months. Now, 8 months to get what's equivalent to about 1.5 PhDs. It's not bad going in terms of time scale, but 8 months is a lifetime if you're sitting on a recall. Bear in mind, these things can be sorted out, but it may take time. It'll be sooner now than it was then because of what we've learned of this particular problem. So I'll just pause while you maybe want to take a screenshot of these papers. Most of them are open access, so if you can't get hold of them, drop me a line. The best one to give you an overview is number 3, and that is certainly open access in JIOAC International. And again, Karen has kindly put together some of the stories at the time. There's Greg Corbishley mentioned on the middle of the right of your screen. And what he did was superb. Get a list, look at it, see what you don't know about, investigate. I think, yeah, there's that one. And so that generated quite a bit of publicity because it was the first time that a recall had been rescinded in the UK. The next surprise I'm going to think about, I invite you to think about, is in relation to Manuka honey. And we're going to have to delve into the technique of stable carbon isotope ratio mass spectrometry, because this is a very powerful technique for authentication of foodstuffs. The reason it's powerful, well, it's based on a natural phenomenon where different photosynthetic pathways fractionate carbon-12 and carbon-13 in different ways, so their ratio changes. And the ratio gets more negative for C3 honeys, C3 sugars here, than for C4 sugars. And honey is sugars, so it can be adulterated by cheaper sugars. And the C3 plants constitute about 90% of all plants. So there's a fair chance, and it is found empirically, the nectar gathered by bees to make honey is overwhelmingly from C3 plants. Usefully, maize and sugarcane are C4 plants with different photosynthetic process, and so the fractionation is different, and so we can separate. Now, why does this technique work so well? Part of the reason it's based on international standards. It's a technique, mass spectrometry, with capable of a wide dynamic range and low dispersion of results if carefully used. More importantly, I think, though, is that multiple communities of scientists use it. The geochemists use it, food authenticity people use it, and forensic scientists use it. Remember the case of the torso in the term Thames that was partially sorted out by carbon isotope ratio mass spectrometry, or generally isotope ratio mass spectrometry. Now, the subtleties of the ways in which the photosynthetic pathways are exemplified by the fact that it can depend on the partial pressure of carbon dioxide in the leaf of the plant in the atmosphere. So quite subtle effects are at play here, but the differences are there to be seen and can be exploited analytically. And so when consignment of Manuka honey was stopped at the UK border, the public analyst was invited to do an analysis, and they reported that it appeared to be adulterated with added sugar. And it was referred to the government clinic. So what I did was to plot all of the data, both the public analyst data and the data supplied by a good laboratory in New Zealand supporting the manufacturer. And the circled results on the left are the public analyst results. And you can see that they're not out of line with the results obtained by the other laboratory. And the distinction between the two sets of results is that at the top, this is the the ratio of the raw product, and the little red crosses at the bottom are the ratio of the nitrogenous component of the honey. Now, the nitrogenous component is used in internal standard for this method, because it's been found that if you refer to the internal standard, the carbon isotope ratio of the nitrogenous fraction, which can be separated from honey, and that provides an added layer of quality control. And on the right then, what we've got is both the bulk honey, and then the little square boxes are the nitrogenous part, the carbon isotope ratio, nitrogenous part. Then there are also data derived from the separated individual fructose, glucose, and disaccharide components of it. And the circled set of results are what it should look like. These are what international agreed limits are for all those different ratios. That's what it should look like. And of course, the results don't look like that. And you can see that some of by and large, most of the carbon isotope ratios of the bulk honey and its separated sugars are shifted to less negative domains. But more importantly, the internal standard, which is supposed to be constant, has shifted to more negative domains. And that makes the amount of apparent added sugar much higher. Not because it's necessarily added, but because the internal standard has shifted. And that's hypothesized to be caused by subtle chemical changes in the reaction, which causes Manuka honey to have its characteristic inherent antioxidant potential. The conversion from dihydroxyacetone, which is got from the nectar gathered from the sperm plants, into methyl glyoxal. Now that potential explanation hasn't ever been challenged, and so it's in the peer review literature, and therefore we advanced that as an explanation, concurring with the opinion of New Zealand authorities. So this particular honey was allowed into the UK. So this is an example of a highly reliable technique, standardized and collaboratively trialed at an international level, which can still give questionable results in certain specific circumstances, which we need to know about. And again, Karen has put together some stories about Manuka and honey authenticity or non-authenticity generally, up to 2016, and some other story from May earlier this year. So it's a continuing, highly salient issue in relation to that. Okay, moving on then, and I'm nearly through. I want to talk a bit about bioinformatics. Bioinformatics, as you know, that's the science of the estimation of and the databasing of the DNA sequence of organisms, the genome. And this relates to a sample of squid, which was labeled, that was produced in New Zealand and packed in the UK from arrow squid caught in Southwest Pacific Ocean. Arrow squid is either Notosudaris goldii or Notosudaris slowii. The public analyst reported the DNA extracted was consistent with that of Ilex argentines, a different species caught in a different ocean. How did the public analyst do it? Well, they extracted DNA and sequenced, and the sequences were compared to publicly available databases of the sequences of a wide range of plants and animals. And the software associated with those databases gives you hits in percentage coverage, how close the match is. But an equally proficient DNA laboratory analysed the defence portion and found that their portion exhibited extractable DNA characteristic in databases of the claimed Nucleodaris species. So Malcolm Burns and his colleagues in the Laboratory of the Government Chemist looked at this and looked at differentiation of these different species by the 16S ribosomal RNA sequence and a cytochrome oxidase 1 gene sequence. And both of these sequences are available in bold, and both, as you can see from the phylogenetic trees on the left, will separate Nucleodaris and Ilex. So when primers and probes were established and used to amplify the extracted DNA from the sample, that extracted sequence was then uploaded into comparison with the databases, the Barcode of Life and the NCBI databases. And both of those returned probable hits showing that Ilex and Nucleodaris were most probable species with high similarity with the target sequence. In other words, the two separate species could not be distinguished. Now, why was that? In a technique which is usually highly reliable with regard to species identification. When the molecular biologists and the team looked into this, they looked at the number of records in relation to these species in databases, and they were low. Moreover, when they looked more closely at the original published papers, which underpin the uploaded sequences in the databases, it is possible that so few of the original individuals from those species were actually synthesized, sequenced, that it may have been that there was a morphological mistake in the taxonomy of this sample that was analyzed to derive the uploaded sequence. And that is a problem with particularly the plant kingdom in relation to bioinformatic databases. This paucity of consistent data for low sample numbers. Going back to the Alm Mahal, we found thousands of records in the databases for almond, for instance, but less than 50 by comparison for Prunus Mahal. And that's an issue around which we published back in 2016 in relation to allergen analysis. So talking about allergen analysis, this slide, I show this one because I'm fond of it. I drew it myself. I like the colors, and it shows the complexity of allergen analysis. It also shows that ELISA is still and will be for the foreseeable future the workforce technique complemented by PCR and complemented when you do it by NCMS. Now, why is allergen analysis particularly challenging? And it's simply because allergens, food allergens, are huge protein molecules. Typical ARA-H2 peanut, 65,000 Daltons. Compare that with several hundred gram molecular size for a typical small molecule aflatoxin, pesticides, veterinary residues, and you can see that the challenges facing analysts are huge in relation to these big molecules. And it wasn't for ELISA using antibodies, we wouldn't be able to detect and quantify allergens with allergens at all. Certainly 10 years ago, before PCR and NCMS was developed. So if we can't work on the challenges of analysis, and work is being done, it's underway, but we're not there yet. What do we do? Well, in critical situations, we should use more than one approach. So if you're investigating a fatality or a big recall or some critical problem, don't rely on one set of results for the allergen. Trends are much more useful, but at least two ELISA platforms. And as my good friend Dionysius from SGS said to me a while ago, not just two ELISAs, but ELISAs that use two different antibodies. So that the problem of cross-reactivity may be reduced. You might also think about using ELISA and PCR, or any combination of two or more of ELISA, PCR, and SMS. Allergen analysis is hampered by the lack of reference material, so there are two sets of peanut and some of the common allergens from NBC, several from NIST, the National Measurement Authority in America, the USA, and Monica and international organizations. So there are some reference materials available, but not enough. And you need to think about how best to use them. And even when there are reference materials, if the target sequences, the amino acid sequences in this case, are different, then reference material may not be as much help as you think. I mentioned the bioinformatics still needs developing, but one thing we can do in the analytical community ourselves now is to report our results in sufficient detail to assist risk managers and risk assessors. And as an adjunct to that, we need to think about whether we're looking at the upper bound or the lower bound of the measurement uncertainty. And again, we can talk about that if we have time. But what do I mean by reporting in sufficient detail? The report must show how it was done. It's done by ELISA, it's done by PCR, it's done by SMS, or whatever combination. And what approach, what kit, what manufacturer, what published paper you're relying on. Then we get a number and a unit. And the number is X. Read this out. And the best estimate of the concentration of allergen found in analysis of the sample received after in-laboratory allergen extraction analysis by molecular method. And those of you who work in a lab will know the importance of that particular phrase, that particular sentence. And then Y is either the allergen protein or the name of the food. Now, milligrams per kilogram, please, not ppm, but milligram per kilogram as either peanut protein or peanut. And why the either or? Well, the protein is the hazard, and the protein is what's determined and reported in dose response curves from clinical studies. Not the food, but the food is what's mentioned in legislation. So many ELISAs report in the food quantity, peanut, milk, almond, and that to get to the protein, you need a conversion factor. And so if you're using a conversion factor, where did you get it? What is it? And what should be in the report as well? Because it depends also on the calibrator as well, what the quantitative result is. As a matter of routine, when you're sick of the sight of it, you should always say, page 1 of the report, page 2, page 10, X milligrams per kilogram as peanut protein or as peanut. You never leave the number without units and what the units mean. Something else, plus or minus BU, plus or minus the expanded measurement uncertainty. Because in allergen analysis, that would be bigger than you think, would be bigger than most of the instances you see in small molecule analysis. And you may have to take that into account. And accredited laboratories will have that information, but they don't usually report it, nor do they report the recovery, which they'll also have. And there are many good reasons for that. I don't want to go into now, but that's another debate I think we should have. So why do laboratories get it wrong? Inadequate planning for sampling, incorrect sampling, loss of chain of custody of the sample. I haven't given you examples of that, but I've come across a few. Inadequate methods that don't address the real issue, inadequate application of the method, inadequate interpretation or reporting, which may have touched on a bit in or nature of drainage springs, or inadequate bioinformatics. These are the things that I suggest you think about if you've got a puzzling result and talk to your laboratory about. So, having said that analysis is not the be-all and end-all, and seeing you the results, I'd now like to pass over to Professor Louise Manning, who will tell you how to do all this without analysis necessarily. Over to you, Louise.
Thank you, Michael. So I'm going to talk quite briefly around procurement and procurement-focused prerequisite programs. And as Michael said in the poll, these were cited as part of a food safety plan as being really important. So Michael, could I ask that you go to the next slide, please? So one of the key aspects of developing your prerequisite programs around your supply chain is mapping the supply base. And when I talk about mapping the supply base, it's simply, as you can see, looking at all your suppliers and where they are, not only your first tier suppliers, second tier, third tier. It may be that you have some very complex chains. Once you've put where they are on a map, you can then start to think about what are the geographic vulnerabilities, the political vulnerabilities, economic or social, how are those changing? And to start thinking about which are the really important components of your products. We've seen vulnerabilities in supply chains for both services and manufacturing aids such as carbon dioxide as equally as individual ingredients which are a strong component of the product, or particular types of packaging. So map your networks, identify where your ingredients, packaging and services are coming from. Consider which materials are also being supplied to your direct suppliers, and then start to think about what potential food safety, food quality, or food fraud risks are in place and how you manage that risk. If we're looking at food fraud, we might talk about intentional substitution, where you think you're buying one material or ingredient and somewhere along the supply chain, illegally, there has been intentional substitution. That's a potential risk that you have to consider. But when you're thinking about your supply chain, you also have to think about substitutability. So during product development, or when you are manufacturing a product, think about, in the event of a major vulnerability, a major earthquake in a particular country where there's a particular ingredient that you are purchasing. We're seeing severe flooding in many countries at the moment. What is the potential for substitutability of those key ingredients? Can they be purchased from other areas of the world? Or because you are so vulnerable, you have to keep a certain level of stock of that ingredient? And these are all the aspects of developing your procurement prerequisite program, so you are clear on where your vulnerabilities lie. Michael, could you go to the next slide? We seem to have gone on to, perfect, thank you. In your procurement activities and thinking about the prerequisite programs, it's important to think about those pre-contract activities, planning. Michael, we seem to have switched again, I'm not sure. Planning, what analysis you may do, what sourcing audits you may do before you actually sign the contract. Post-contract, how you continue to develop contract management, how you develop supply chain management protocols, and also think about disposal of materials, ingredients that may have come from other parts of the world, which could present environmental or legislative issues in the country where you're actually using them. And there's all those other general activities that sit around procurement. What is your corporate governance structure? How does supplier relationship management work across your business? Is risk management undertaken within the technical department and also more widely? What particular vulnerabilities are on your corporate risk register that may sit with senior management? And how are you ensuring regulatory compliance as regulations may change? There's a whole range of risk mitigation strategies that could take place. And you're going to get the slides afterwards so you can look at them in detail. But are you including your suppliers in your risk planning and risk mitigation? Are you working with them to look at potential supply risks that could occur and how you are going to manage that? Have you considered insurance for particular vulnerabilities that you may have? How are you on an ongoing basis going to work on your product and service specifications? Can you work with your suppliers to think about substitutability and how you may do that in practice if there was a major event in some part of the world? Thinking about diversifying your suppliers to minimize risk of a non-supply situation of a key ingredient or material. And also thinking about how you embed your procurement due diligence procedure. Could we go to the next slide, Michael? So that's all I'm going to talk about today. I'm now going to hand on, I think it's to Charlotte, who's going to talk through the next few slides. So thank you for listening.
Thanks, Louise, and sorry about the glitches. I forgot I was still screen sharing for a second there. Just before Charlotte comes on, I just wanted to point out some IFST resources. In particular, there's a whole suite of information statements, and you see the list there on the right of the screen in relation to sampling and analysis. And the link is there, but if you go into IFST and look for science and technology resources, you'll find lots of information on how to do sampling and analysis correctly. And then there's also, if you're not aware already, the 3 knowledge hubs, one in relation to COVID, one in relation to food allergens, and one in relation to sustainability. And each of these knowledge hubs contain attested and trusted resources, very useful if you need a way into the problem that you're handling. I just have to finish with acknowledgements of all these people who've helped me over my career and some of the funders who funded some of the studies I've been talking about. And now I would finally like to hand over to Charlotte, and I'm stopping sharing now.
Brilliant. Thank you, Michael and Louise, for a great presentation there. Michael, I think this one probably for you. So do you have any advice on identifying issues, sampling and ensuring transport vehicles? Or maybe Louise in that one.
Let me, if I can do that one first. Not my field directly, but dedicated transport, if it's perhaps a luxury. But if you're worried about commingling of agricultural material in relation to allergens, then it's a real issue. And make sure that you clean and sample and swab the outlet of the container. So if it's a tipped container or it has a suction or gravity fed connection, it's often there that you'll find the previous cargo having gathered. There are also, even in tip disposal transport vehicles, you find dead ends and nooks and crannies where the previous consignment can gather and then go on to contaminate a subsequent one. So it's really like looking at sampling, cleaning in place and sampling in a production line. You look for the dead ends, you look for the places where hang-ups can occur, and you make sure they don't. and you make sure that you sample for them or swab them when you get into it. Is that a sufficiently have we got away with that one, Louise? What do you think?
Yeah, no, I think that's a really good advice. I would also say in your supply chain mapping, think about how people are changing their logistics practices with regard to reducing their carbon footprint, because many businesses are looking to backload and take other products back to on the reverse journey. Others are thinking about how they can use their vehicles for multiple uses, for example, moving food waste, and all of these things have a potential food safety challenge. So as you're looking at developing your strategies to reduce carbon footprint, think about the implications for food safety as well, because it's really important.
Brilliant. Thank you both. Well, there is another questions on the list, but we're running out of time. So what we will do is, if there are any more questions coming through, please do pop them in the chat. We will answer those, and we will publish an FAQ document on our IFST community page. So do look out for that, and we will get that on there as soon as possible for everybody alongside the recording. So I'll hand over to Peter now to finish us up.
Fantastic, thank you very much. And Robin, if you would like to just relaunch the poll, just to see if we've got anything that's fantastic. So whilst I thank Louise, Charlotte, and of course Michael for the excellent presentations this afternoon, thank you all very much to everybody for attending this afternoon's webinar. There will be a recording of this webinar shared later, which will include this Q&A session. And that, as Charlotte said, will be hosted through the IFST Community Hub. If you've all answered that poll, Robin, if you'd like to pull up those results, and we'll just see if that's changed anything from earlier. So we have 0, 0. Ooh, we have had a shift. We've had a it feels like Eurovision all over again. So auditing's got nil point, as is documenting. On our previous poll, you'll be pleased to hear, Michael, that testing got 6% of the votes. Now it's got 19%. So you may be coming back to work yet. HACCP has actually gone up. HACCP's got, and PR prerequisite programs have gone from 61 to 65%. And that's all at the expense of training and awareness, which is interesting. Which previously was 33%, has now plummeted to the very depths of 15%. Oh, as you can see, Robin has done fantastically at pulling that information through, which is brilliant. Well done, Robin. That's brilliant. So yeah, so thank you again very much for everybody attending this afternoon. Hopefully you found it interesting, where we've looked at some of the previous incidents of food problems that have occurred, and hopefully being mindful or cognizant of those challenges, we'll actually be able to use our crystal ball to avoid those sorts of problems and falling into those traps again, which just leaves the curveballs that seem to come at us from all directions, particularly at the moment, to come and surprise us at 4 o'clock on some idle Tuesday. So again, thank you all very much. Before you log off, there will be a short survey that Robin will launch as the webinar ends. Thank you again to Louise, Charlotte, and Michael, and have a good rest of your afternoon. And thank you again. Goodbye.